Hello, everyone, and welcome to today's webinar, HIV-1 Genotyping Targeting Integrase, Perspectives on Surveillance of Antiretroviral Drug Resistance. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com/HIVDR. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. In addition to submitting questions, we ask that you participate in poll questions during the presentation and a survey at the bottom left of your screen. To submit your answers, click on the answer that is most accurate for you. This webinar is educational and thus offers free continuing education credits. Please click on the continuing education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speakers. Dr. Kim Steegen, Senior Medical Scientist, Charlotte Makeke, Johannesburg Academic Hospital, Johannesburg, South Africa. Dr. Natasha Wood, Co-founder and Chief Science Officer, Hyrax Biosciences, Cape Town, South Africa. And Dr. Steve Williams, R&D Leader, Capillary Electrophoresis, Thermo Fisher Scientific, South San Francisco, California. Dr. Steegen, you may now begin your presentation.
I would like to thank Thermo Fisher for giving me the opportunity to present on this webinar, and I will be talking to the global status of HIV drug resistance. By 2020, about 37.7 million people were estimated to be living with HIV, including 20.6 million people from Eastern and Southern Africa, which represents the majority of the epidemic. Antiretroviral rollout has started in 2000, and by 2020, about 27 million people were on ART, which is a threefold increase since 2010. The massive rollout of antiretroviral treatments has also significantly had an impact on the decline of AIDS-related deaths. In 2016, the UN set up some goals in order to have as many people living with HIV to know their status, of those that know their status to be on treatment, and of those that are on treatment, the majority being virally suppressed. Although those targets were not met by the end of 2020, we were not far off. 84% of people living with HIV know their status. Of those that know that are HIV positive, 87% are on treatment, and of those that are on treatment, 90% are virologically suppressed. HIV is a virus that has a very high genetic diversity. This diversity is driven by the production of 1 to 10 billion virus particles per day, and in addition to the reverse transcription process being prone to error. The reverse transcriptase enzyme does not have a proofreading mechanism to correct for the mistakes, and therefore we expect about 1 random mutation per new provirus. The higher the levels of viral replications, the greater the risk for the development of drug resistance, as more replication means more mistakes are being made. Virological suppression is not only important to reduce the risk of transmission, but also to reduce the risk of resistance. Therefore, we need efficacious antiretroviral treatments. When we look for efficacious antiretroviral treatments, we look for drugs that have a very high viral potency, have a very high genetic barrier to resistance, and in addition, good safety profiles, limited drug-drug interactions, and the possibility of co-formulation between different drugs increase the adherence in patients and therefore increases increases viral suppression. In this figure, the HIV life cycle is represented with the different steps in the life cycle, and it's indicated which different classes inhibit the different steps in the life cycle. The most commonly used drug classes are those that inhibit the reverse transcriptase step, including the NRTI and the NNRTI drugs, as well as the protease inhibitors. And more recently, the integrase inhibitors have been included as well. Initially, the integrase inhibitors were mainly focused in resource-limited settings to treat patients on third-line treatments, whereas with the introduction of dolutegravir, there has been a shift of using integrase inhibitors in first-line treatment. However, even though some of the newer drugs have a very high genetic barrier and are very good drugs, all available drugs, including the newer classes, are at risk of becoming partially or fully inactive due to the emergence of drug-resistant virus. HIV drug resistance can reduce the effectiveness of antiretroviral treatment, not only on an individual patient level, but in a global perspective as well. Therefore, it is important to monitor HIV drug resistance, especially in regions where HIV drug resistance testing is not available at patient level. The outcome of these surveillance efforts can then inform public health treatment guidelines. We would now like to participate in the first poll for this webinar.
So our first poll question is, do you perform HIV drug resistance surveillance testing in your laboratory today? The options are yes, no, but we are planning to add it to our test menu offering, or no. Please click on your selection to record your answer. And I'll give you a short time to make your selection. And I'll turn it back to Dr. Steegen.
Thank you for your participation in the poll. The World Health Organization has set up an HIV drug resistance strategy, and they report on a regular basis on the outcomes of countries that have performed drug resistance surveillance. So in the next couple of slides, I will touch on a couple of the results that were published in the most recent 2021 drug resistance reports. There are four main pillars in the drug resistance strategy from WHO. The first one is focusing on pre-treatment drug resistance in adults initiating or reinitiating first-line treatment. The second one is pre-treatment drug resistance in ART-naive infants who are newly diagnosed with HIV. Number three is acquired drug resistance in adults, children, and adolescents. And finally, surveillance of patients who are on pre-exposure prophylaxis who are diagnosed with HIV. I will discuss some of the results for the first three pillars. I will, however, not touch on results from the last pillar. Starting with pre-treatment drug resistance in adults initiating or reinitiating first-line treatment. 21 out of 30 surveys reported first-generation NNRTI pre-treatment drug resistance levels above 10%. This is indicated on the graph on the right with the vertical red line, which indicates the 10% cutoff. All countries that reported than 10% do have dots on the right-hand side of the bar. So you can see that NNRTI drug resistance is quite significant in most countries. In patients initiating or reinitiating treatment. Overall, the prevalence of first-generation NNRTI pre-treatment drug resistance was 3 times higher among people reporting previous ART exposure, being on average 24%, compared to those who are truly ART naive, being 7%. The prevalence of pre-treatment drug resistance overall is still driven by NNRTI resistance, which is indicated in the red box. On the other hand, NRTI resistance in the amber box remains relatively low, and if we look at PI pre-treatment drug resistance, it's even lower, as for the same with second-generation integrase resistance. Based on these results, there has been a change in many treatment guidelines, including the WHO guidelines, where the preferred first-line treatment regimen has been changed from an NNRTI-based regimen to now a combination of tenofovir, 3TC, and dolutegravir, which is offered in a fixed-dose combination, also referred to as TLD. Moving on to pretreatment drug resistance in ART-naive infants who are newly diagnosed with HIV. There were surveys conducted between 2012 and 2020 in about 10 countries, and NNRTI resistance was observed in over 70% in some of the countries. Overall, across all the countries, at least half of the kids born HIV positive had at least one drug resistance mutation. Although the level of NRTI resistance was a bit lower, it still exceeded above 10% in most of the surveys conducted, meaning that there is quite a bit of transmitted resistance between mom and child. Based on these results, the guidelines have changed once again, whereby WHO initially recommended a PI-based regimen, moving on from an NNRTI-based regimen, but now with the introduction of dolutegravir, the preferred regimen is actually a dolutegravir-based regimen. Finally, moving on to the acquired drug resistance in adults, children, and adolescents. So this is when surveys are being done in patients that are on treatment but are failing at the time of resistance testing. Surveys are usually conducted at 12 months and then at 24 months of treatment. In this graph, it is obvious that most patients failing first-line NNRTI-based treatment have a very high prevalence of resistance, in some countries over 90%. When we look at surveys done at patients that have been on treatment for 48 months, the level of resistance drops slightly, but there is still a very common presence of resistance in these patients. So based on these results, it is once again obvious that NNRTI-based ART has a very low genetic barrier to resistance, meaning that many patients failing these regimens present with drug resistance. Therefore, the second-line treatment guidelines have now been changed. For adults currently failing non-DTG-based ART, they should be switched to a DTG-based regimen with an optimized NNRTI backbone. And in addition, adults that are currently failing a first-line DTG-based regimen, they should be switched to a boosted PI-based regimen. Among the surveys that were conducted and that were reported in the WHO report, only 10 out of 35 countries included integrase resistance surveillance. In these surveys, less than 0.4% of drug resistance was detected, which is extremely low. We do, however, need to take into account that the rollout of DTG so it is very common that we don't see a lot of resistance. Therefore, WHO recommends to include INSTI resistance testing for all new surveillance activities going forward. Switching gears a little bit now, so we're now no longer talking about the WHO reports, but because there is so limited information available on integrase resistance, I thought it would be informative to also look at what is happening in the Western world. In this study in the US, they looked at 28 jurisdictions between 2014 and 2018, included more than 50,000 people, of which almost 20% had at least one drug resistance mutation. Despite that the rollout of integrase inhibitors has been much longer compared to low and middle income countries, they still only see a 0.8% prevalence of integrase resistance. And the resistance in pretreatment patients is still driven by NNRTI resistance. In a recent review, they looked at acquired resistance of patients failing DTG-based treatments, and they found that the risk factors for DTG resistance were mainly poor treatment adherence, drug interactions, and HIV disease factors such as high baseline viral loads and active opportunistic infections. In the review, they only included 15 reported cases of dolutegravir resistance because dolutegravir resistance is so rare. Of those 15, 10 had either suboptimal DTG levels or poor adherence. 3 patients had advanced HIV and severe active infections, and 4 treatment-naive and 5 treatment-experienced people had a baseline viral load of over 100,000 copies. There is, however, a concern that the limited access of viral load and genotypic testing in low and middle-income countries may influence the proportion of people with virological failure and therefore the emergence of integrase resistance. In Canada, a surveillance was done between 2009 and 2016, including close to 10,000 individuals, and they found that the level of protease and reverse transcriptase resistance decreased significantly from 337 cases per 1,000 treated individuals in 2009 to 285 per 1,000 treated individuals in 2016. On the other hand, the integrase resistance increased significantly but remained very low. It went from 1 per 1,000 treated individuals in 2009 up to 7 per 1,000 treated individuals in 2016. Of the 64 individuals that were newly identified as having integrase resistance, the majority of them were driven by exposure to raltegravir, which is a first-generation integrase drug, whereas there were only 3 cases of new integrase resistance mutations for patients with DTG treatments. So to date, most surveillance efforts were focused on the protease and reverse transcriptase gene, but with the massive rollout of dolutegravir and possibly other integrase inhibitors, WHO recommends to include integrase resistance in any new surveillance activities. In conclusion, HIV drug-resistant surveillance has informed policymakers on the best treatment regimens in certain populations, especially in regions where a public health approach is required. As countries scale up the transition to integrase-based antiretroviral treatment, surveillance of the integrase gene, especially in low and middle-income countries, is required. This was the end of my presentation, and I invite you to participate in the next poll. Thank you for your attention.
For our second poll, the question is, for what applications does your laboratory perform HIV-1 genotyping? Please select all that apply. Your options are clinical therapeutic management, epidemiologic and/or genetic surveillance, or we do not perform HIV-1 genotyping today in our laboratory. Again, remember to click on your answer. You may select more than one for this poll question. And next, we will be hearing from Dr. Wood.
Thank you very much, Dr. Stegen, for the informative presentation. And hi, everyone. Thank you for joining. As I've been introduced, my name is Natasha, and I'm from Hyrax Biosciences, which is a company that develops software for DNA sequence analysis. And today, specifically, I'll be talking to you about our X-Type Sanger HIV product, which is a platform for HIV drug resistance analysis and surveillance monitoring. So just as a presentation overview, I'll first provide you with a brief introduction of the X-Type workflow. I'll then jump into a series of screenshots to show you exactly how X-Type works. And then finally, I will conclude on how Thermo Fisher and Hyrax Biosciences are supporting this global drive for increased surveillance. So as I've said, X-Type Sanger is an online platform which you can access at the URL at the top left. And it is an automated HIV drug resistance reporting platform for Sanger sequencing data. And to place it more in the context of HIV genotyping, if you think of HIV genotyping as a 3-step process, the first step is to do sample prep, where you extract your DNA or RNA. The second step is the sequencing, where you generate your data. And the final step here is the analysis, where you actually do the interpretation and the analysis of the data. And this is where X-Type comes in. And we really think about Exotype as an out-of-the-box solution, which consolidates the global best practices for HIV Sanger data analysis, specifically for HIV. So what Exotype does is base calling and editing. It does consensus sequence generation, quality control analysis, drug resistance reporting for surveillance. The results are available for download in multiple formats, and you can share your results between peers within an organization. And if you're wondering what the underlying programs and software are that power Exotype, we've really consolidated the global best practices. So on the left over here, you can see your AB1 files, which come from the sequencing instrument. And in the next image, we have licensed the University of British Columbia's Recall software to do chromatogram editing and review. So when you see this online on the user interface, you are actually using the Recall software, which you may be familiar with. Then finally, on the next image, the Stanford HIV Drug Resistance Database, we have implemented this algorithm in our software as well. And just a note on this, when Stanford updates the drug resistance database and the drug calls and resistance scoring, we update it on our side within 48 hours. So you're always running the most up-to-date software on Exotype. And then finally, on the right, you can see that you get your drug resistance reports and drug calls for surveillance and research. And we also provide these quality control and surveillance monitoring results, which have been laid out by the WHO in this document shown over here, which consolidates all of these best practices. So that, in a nutshell, is an introduction to Exotype, and I'll now take you through a series of screenshots. So once you have logged in and created an account, you will access the dashboard over here. To run your data, you create a new job or a new analysis, and you go through a series of steps. Over here, I've shown you the first step, which is just to create the job. And you can see at the bottom left, you need to select if you're running the HIV genotyping kit with Integrase or without Integrase. And these are provided by Thermo Fisher. So once you've gone through the series of steps, which you can see in the tabs on the top, so job details, select data, all the way to review and submit, which is the final step. Once you click this, you will then be taken to a results page or a pre-results page. And here you can see on the left, in orange, you've got all the samples that you've uploaded. And for each of these samples, you then have an associated chromatogram file. For each sample, if you've run both protease and reverse transcriptase, as well as Integrase, you will have a separate chromatogram for each of these gene regions. And as you are more familiar with than I am, I'm sure you will be able to do your chromatogram editing and review for each of these gene regions and each sample. Once you've completed these steps, you will then have your results. And the results include quality control overview. And over here, you can see the results of the sequencing control analysis. You also then have sample-specific results, which include the sample warnings, as well as drug calls and drug scores for surveillance. And then finally, you have a series of downloads that are available for downstream analysis, which include your consensus sequences in different formats, as well as a detailed summary file, which is a tab-separated file that you can open in Excel. And this file is particularly useful for your surveillance, large-scale surveillance studies. So I know I've just shown you a few screenshots here, but I would like to encourage you to please get in touch if you would like a demo or a training session, and you can get in touch at sanger@hyrexbio.com. So please don't hesitate to get in touch for a more in-depth overview. And then just to tie it back into the surveillance studies, there are several indicators for surveillance studies, and one of these are the sequence similarities. So if two viral sequences are too similar to one another, this may indicate specific transmission chains. So reporting on sequence similarity is very important to monitor your transmission dynamics. To show you how Exotype does this, in the right-hand side image over here, you can see in orange that 4 samples have been flagged as too similar to one another. So D8, D6, D9, and D12 are too similar to one another. There are too few differences between their nucleotides. And if you then plot this on a phylogenetic tree, you can see that these 4 samples cluster on a single branch, which indicates that they may have been transmitted as a cluster. So before I hand over to Dr. Williams, who will take you through the sample prep and the sequencing steps, I'd just like to conclude that as HIV genotyping scales across the world, these kinds of streamlined laboratory methods and sequence analysis tools are really key to facilitate the ongoing surveillance of drug resistance mutations. And this is especially in light of Dr. Stegen's presentation, which presented the WHO drug resistance report, and it highlights the challenges of implementing these national surveillance plans. And then finally, again, I would just like to encourage you to get in touch for a demo or a training session if you are interested. And thank you very much for your attention.
Hi, thanks for joining me today. I'm Steve Williams. I lead the assay development group within the clinical testing solutions business at Thermo Fisher Scientific. And today I'd like to describe the design and the performance of the Applied Biosystems HIV-1 Genotyping Kit with Integrase that is able to detect HIV drug resistance mutations relevant to the most widely used drug classes across a broad range of viral subtypes. As you may know, Thermo Fisher launched the first iteration of this product, the HIV-1 Genotyping Kit, in 2017, and it's been widely used to help survey drug resistance for protease and reverse transcriptase inhibitors in low to middle income settings. The new genotyping kit that we're introducing retains the same excellent performance characteristics of the original kit, but adds additional sequencing of a region of the integrase gene as the use of the integrase inhibitors expands in low to middle income countries. So specifically, the HIV-1 genotyping kit with integrase detects resistance mutations in protease, reverse transcriptase, and integrase. It is a cost-effective solution and has broad coverage of all of the HIV-1 group M subtypes, including the circulating recombinant forms of the virus. The kit is compatible with plasma and dried blood spot sample specimens, and it has a scalable workflow that can deliver a drug resistance result in about 14 hours, all the way from RNA extraction through a drug resistance report. And that workflow represents less than 3 hours of total hands-on time. The kit comes with pre-mixed reagents to minimize the number of pipetting steps, and includes a number of controls in case of the need for troubleshooting. And the analysis of the Sanger sequencing trace reactions can be performed on a series of different genetic analyzers that represent different throughput requirements. And then finally, as Natasha explained in the previous slide, we have a easy to use software to interpret the Sanger traces and to identify the drug resistance mutations. So the assay workflow is shown in this slide, beginning with the specimen processing to extract the viral RNA. After RNA extraction, there is a RT-PCR step to amplify the virus, followed by a second amplification step that's a nested PCR reaction, and then Sanger sequencing of the drug resistance regions and interpretation of the Sanger sequencing traces is performed. As mentioned previously, the end-to-end process takes about 14 hours with about 3 hours of total hands-on time. The RNA extraction can be performed using the MagMax viral pathogen kit from Thermo Fisher, although any extraction method yielding high-quality RNA is acceptable. The MagMax protocol has been optimized for both plasma and DBS samples. And the genotyping kit itself consists of 2 different modules. The first module is an amplification module that does RT-PCR followed by nested PCR. And the second module is the sequencing module. Importantly, the first amplification step can amplify all three regions of the virus, protease, reverse transcriptase, and integrase, in a single step. So there's no need to split the extracted RNA sample into two different amplification reactions. The second nested amplification step does require splitting the PCR reaction into protease and RT nested PCR and also integrase nested PCR. And then the second module in the kit consists of all of the sequencing mixes that are required to sequence the amplicons prior to running capillary electrophoresis to separate the Sanger fragments and call bases. And then the final step in the workflow is the assembly of the individual sequences into a consensus sequence by the Exotype software and calling of the presence of any drug resistance mutations. Drilling down in a little more detail, the HIV-1 genotyping kit amplifies the protease, reverse transcriptase, and integrase regions, which can predict drug resistance for nucleoside reverse transcriptase inhibitors, for non-nucleoside reverse transcriptase inhibitors, protease inhibitors, and integrase inhibitors. In the PR region, codons 6 to 99 are amplified. In the RT region, we amplify codons 1 through 251. And in the integrase region, codons 1 through 288 are amplified. So the first step, as I mentioned, of the amplification is a multiplex RT-PCR, where both protease, RT, and integrase are amplified. And this produces two different fragments, which are further amplified in a nested PCR protocol. For protease and RT, we produce a 1.1 kilobase fragment, and for integrase, it's a 1 kilobase fragment. So following amplification, the success of the amplification is confirmed by running a gel and looking for a band of the correct size. At this point, if the amplification is successful, you proceed on to the sequencing step of the workflow. The sequencing is performed with, in the case of protease and RT, 6 different sequencing primers to cover the entire length of the nested amplicon. These are 3 forward sequencing primers and 3 reverse primers. And in the case of integrase, 4 different sequencing primers are used to cover the amplicon length, 2 forward primers and 2 reverse primers. The result of the sequencing is that for any given nucleotide in either of the amplicons, you have at least 2x coverage, both a forward and a reverse read for each nucleotide. And following sequencing, the reads are assembled by the Exotype software, and the consensus sequence checked against the Stanford HIV drug resistance database. So the performance of the HIV-1 genotyping kit was validated in collaboration with the CDC in Atlanta with our colleagues. And as part of the validation, the performance was assessed according to the WHO validation criteria, where we looked at subtype coverage, accuracy, amplification sensitivity, precision, and reproducibility. So briefly, this validation procedure starts with either 200 microliters of plasma or 1 to 2 dry blood spot samples. RNA extraction is performed, and then 10 microliters of extracted RNA is input into this multiplex RT-PCR reaction to amplify the 3 different drug resistance regions of the virus. The RT-PCR reaction, as we've discussed, is split into 2 different nested amplification steps, one to amplify protease and RT, a second to amplify integrase. And then finally, the nested amplicons are sequenced by Sanger sequencing and run out on the CE instrument. So to demonstrate the broad subtype coverage, samples with known subtypes were analyzed in both plasma and DBS formats. In a total of 139 samples were analyzed that were split between 39 clinical research samples and 59 analytical samples. The clinical research samples came from Nigeria, Kenya, and Haiti, And the analytical samples were a mixture of either external quality assessment samples from proficiency panels provided by the CDC, viral isolates, or cultured integrase clones from Stanford. And as you can see there from the table in the slide, there was successful amplification seen across all of the major group M HIV-1 subtypes, including the CRS and recombinant subtypes as well. Next, we looked at the assay accuracy. So the accuracy was determined for both plasma and DBS samples across a wide range of viral load, all the way from the limit of detection of the assay up to quite high viral loads, up to as high as 10 to the 6 copies per mil. The accuracy was determined by comparing data against reference sequences from 3 different sources. Firstly, for the clinical samples clinical research samples that we looked at, the sample was compared against the sequence data from the original Thermo Fisher protease RT kit, or in the case of the integrase region, against the ViraSeq integrase assay. For the proficiency samples that were looked at, the consensus sequence from the proficiency exercise was used as ground truth, and for the isolates, data from Stanford University, sequence data from Stanford University was used. So the conclusion from the accuracy study is that for both plasma and DBS samples, more than 90% of the comparisons between the test result, the result run with the new HIV-1 genotyping kit, and the reference sample were at least 98% identical. So very high concordance with the ground truth in this case. Next, we examined the assay amplification and at the amplification sensitivity. And this was measured by the presence of a band on the gel for the nested PCR product. It was determined for 2 different groups of plasma and DBS samples. The first group had a viral load of greater than 5,000 copies per ml. And the second group had a viral load between 1,000 and 5,000 copies per ml. And for the second group, this was considered at or close to the LOD of the assay. For the plasma samples, we found that more than 90% of the specimens and replicates were positive when the viral load was between 1,000 and 5,000 copies per ml. And more than 95% of the samples were positive, i.e. they produced a band on the gel, when the viral load was over 5,000 copies per ml. For the DBS samples, more than 85% of the samples were positive when the viral load was close or at to the limit of detection between 1,000 and 5,000 copies per ml. And at the slightly higher viral loads above 5,000 copies per ml, more than 90% of the samples produced a band on the gel. So good assay sensitivity for both plasma and DBS samples. We also looked at the assay precision and reproducibility again, across a wide range of viral loads, all the way from, in the case of the precision study, 5,000 copies per ml, and the case of the reproducibility study, 2,000 copies per ml, up to quite high viral loads. For the precision studies, 3 sample replicates were extracted and tested together by 1 laboratory scientist. And for the reproducibility studies, 3 plasma samples at different viral loads were run with 5 replicates on different days by 2 lab scientists. And then pairwise comparisons at the nucleotide level were performed to assess the reproducibility and precision. And in all cases, the pairwise comparisons at the nucleotide level were very, very good, greater than 99% across all of the viral loads examined. So in conclusion, the HIV-1 genotyping kit with Integrase meets all of the WHO HIV-DR assay performance criteria for surveillance. It has broad range of subtype coverage for both plasma and DBS samples, has excellent specificity and a sensitivity of 1,000 copies per ml for plasma samples and 2,000 copies per ml for DBS samples. A range of different capillary electrophoresis instruments can be used depending on throughput needs to analyze the Sanger sequencing reactions, and the workflow enables the user to go from sample specimen to called drug resistance mutation in less than 14 hours with about 3 hours of total hands-on time. And the kit is configured with premixed primers, and all of the kit components have relatively long shelf lives, and of course the kit receives dedicated support from Thermo Fisher Scientific. And for more information on the availability of the new kit, I encourage you to visit the website at the address here, www.thermofisher.com/HIVDR, and you can sign up for information on when this kit will be available commercially. So with that, I'd like to thank you for your attention and thank both our two previous speakers for their efforts. So at this point, I'd like to transition to a roundtable discussion. So Kim and Natasha, thanks for joining me. We'll take some questions and have some general discussion about the HIV genotyping kit. So one of the first questions that we have is around the availability of the Exotype software and how that fits into the overall product offering. So maybe Natasha, I can turn to you and get a little bit more information about that.
Yes, absolutely. And thank you for the question. And sorry that we didn't make it explicitly clear during the presentation. So for anyone who purchases either of the HIV genotyping kits available from Thermo Fisher, what you do when you sign up is you send us a lot number or a purchase number of that kit, and we verify that you are a Thermo Fisher customer. And what happens is that you've got free access to the Exotype software. So you can run unlimited samples using Exotype to do your HIV-DR analysis at no cost.
Okay, great. Thank you, Natasha, for clarifying that. You know, one of the interesting topics for discussion here is around what lessons might be learned from drug surveillance programs, in the United States or in Europe, and how we can apply the lessons learned from both efforts to drug resistance surveillance in low to middle income countries. So, Kim, I'm wondering if you have a perspective on that.
Thank you, Steve, for that question. I think indeed we always need to learn from what has happened in the past. And the fact that dolutegravir has been rolled out for a longer period of time in the Western world is definitely important to consider. As I presented in one of the slides, the level of drug resistance for integrase inhibitors once exposed to dolutegravir remains extremely low, which is a very good sign. However, we do need to be a bit cautious because the monitoring of patients in the Western world using virological failure is more frequent than what it is in low and middle income countries. So we definitely need to keep an eye on it, and that's exactly why surveillance is so important in these low and middle income countries. Because if patients are not monitored very frequently, there is a higher risk that there is virological failure for an extended period of time before it is detected, which then obviously increases the risk for development of resistance.
Yeah, I wonder, I mean, there is a sort of a complexity in monitoring patients in this setting as well. Anything to add regarding the effectiveness of being able to monitor patients in this way?
Are you referring to viral load testing or drug resistance testing?
Really, drug resistance surveillance and testing, yes.
Well, if you compare it to the US, for example, I don't know what the situation is at the moment, but patients might get a viral load up to 4 times a year, whereas in resource-limited settings, it's usually at the maximum 1 time a year if they're doing well on treatment, meaning that a patient in a low-income country might actually be failing for up to 12 months before the first detectable viral load is being confirmed, which definitely increases the risk for resistance.
Yes, yes, thank you. Natasha, anything to add around lessons learned from drug resistance surveillance and testing in the US and Europe?
Yeah, just the comments on the data availability and the amount of information we have for surveillance studies. Historically, a lot of the information has been for subtype B, which is prevalent in the US and Europe, whereas in lower and middle income countries, it's mainly non-B subtype. So it really would be interesting to see how dolutegravir resistance develops in the lower and middle income countries. So that will be an interesting measure to keep an eye on for sure.
Great. One of the questions we had from the audience was around the regulatory plans for the genotyping kit. So as we discussed in the presentations, this is currently an RUO product intended for use in surveillance activities. But I can let the audience know that we are pursuing also IVD status for this product, so that in addition to surveillance activities in the future, the kit could also be used for patient management. So that is something that is actively being worked on, and we'll update on the regulatory status of the product in the not too distant future. I think COVID has really changed quite significantly the structure of virus surveillance programs around the globe as we continue to monitor the evolution of the SARS-CoV-2 virus. And I know in Africa that WHO has actually been working to scale up pathogen surveillance in response to COVID, and that they've set up a network of labs in Africa that are able to produce much higher quantities of sequencing data than previously were possible. But I'm wondering, are there some other key learnings from the ramp in surveillance and how this might have impact as a result of COVID, and how this might have impact on HIV-1 genotyping surveillance efforts? Kim, I'm wondering, do you have thoughts on that?
Yes, as much as COVID has taken over our lives in a negative way in many cases, I think there are also many positive lessons that can be learned from this. And I think there is definitely an increased capability of sequencing in general, for example, on the African continent, and that is largely driven by the need of doing COVID sequencing. And once that capability is there and the instruments are there is skills development, and then it's not so difficult to switch over from one organism to another. The other point is definitely the sequence data availability from COVID. All the COVID sequences are immediately put in a global database. And if you look at the WHO drug resistance reports, as important as the information is, many of the surveys are quite outdated by the time these reports come out. And we're looking at data from 2015, 2016 in the 2021 reports, whereas for COVID, data is available within weeks or within months. So I think there is definitely a possibility that also for HIV, these data could become available much more quickly. And I think that would definitely benefit the surveillance efforts.
Yeah, no, totally agree. Natasha, anything to add there?
Yes, yeah, absolutely. I think COVID has done a lot to improve surveillance globally, and especially in terms of the sharing of information and data between countries and laboratories. And in some ways, it almost goes both ways. A lot of the low and middle income countries have been set up quite well for doing data aggregation because of HIV. But I think one of the good things about COVID is almost the standardization of data. So the way that we can compare COVID data it's really phenomenal. And I think there's been a little bit of a lack of that kind of standardization for HIV, and it would be great to see, like Kim said, just the access become available so much sooner instead of years and years after the data aggregation for HIV. And also looking ahead, in terms of co-infection for HIV patients as well as COVID-infected patients, it will be fascinating to be able to do surveillance on this kind of co-infection.
Okay, yeah, thanks for that. Now, that's a couple of really great things that have come out of COVID in terms of improving, infrastructure and sequencing capability. What about, maybe some of the less than desirable impacts of COVID in terms of, being able to monitor HIV drug resistance? Have we seen, Kim, some negative consequences in terms of the recent COVID pandemic?
Yeah, I think in terms of the surveillance efforts, a lot of the funding that was initially allocated for HIV has been shifted to COVID, which probably delays the current HIV surveillance that was planned or was ongoing. And in addition, more on a patient level, the hard lockdowns have definitely had a negative impact on the frequency of viral load testing, for example. Patients were scared to go to healthcare facilities and were therefore skipping their appointments. And in addition to that, it has also had a negative impact on treatment pickup. And if patients don't pick up their pills, then obviously they are non-adherent, meaning that they have a higher risk of developing resistance, which we will probably only see the impact of this in the next couple of months, if not years, because obviously resistance doesn't develop overnight. But if patients are less adherent, the risk definitely does increase. But with the infrequent monitoring, we will only find out later on which patients are actually not doing so well compared to others. So unfortunately, that has been a bit of a negative thing. On the other hand, some countries have taken that as an opportunity to push, for example, dispensing drugs for 6 months instead of 1 month or 2 months, so that patients actually don't have to go to clinics that often, which I think has, in the HIV world, has been an objective for a very long time, but it would work. And now we were kind of forced to implement these things, and it seems to be working. But again, surveillance will tell us later on how this impact actually has a further on impact on the resistance overall.
Yeah. Natasha, from your perspective, have you seen any more negative impacts of the COVID pandemic on capability to monitor HIV drug resistance?
Yeah, so just a quick comment on that, I guess, is on, from our side, we can see the number of sequences that are being run on a monthly basis. So the statistics for that substantially decreased. It was almost non-existent for about a year. And that's both for just the limits in resources. So a lot of the laboratory staff were reassigned to COVID surveillance during 2020. So there was just a lot less information. So we have started seeing a pickup now. So hopefully patients will be, analyzed, or the data will be analyzed, and the efforts will just be distributed a bit better back to HIV as well.
Yeah. One of the questions that we had from the audience was around the availability of the kit. I think we tried to cover that in the presentations, but just for further clarification, the launch of the HIV-1 genotyping kit with Integrase, the RUO product, is pending, and I encourage everybody to visit the website, www.thermofisher.com/HIVDR, to sign up for updates on product launch and availability. So that is definitely your best source of information with regards to the availability of the product. I want to turn just briefly to different approaches to different technologies that are in use for HIV drug resistance testing. I think, in the US in particular, we have seen a trend towards NGS technologies for drug resistance monitoring. And I'm wondering, Kim, would you mind offering up just a brief comparison between NGS and Sanger-based approaches in the LMIC setting?
Yeah, so it depends a bit what the situation in your lab is. If you have a lot of samples that come in batches, then probably an NGS approach might be more cost efficient and more time efficient as well. However, if you are performing drug resistance testing on samples that come in small batches or like 2 every day, then a Sanger sequencing approach is probably the better option in getting the samples out in an acceptable turnaround time, if you want. From a technical point of view, I'm convinced that if labs that are currently doing Sanger sequencing, that provided they can buy the equipment or get access to the equipment, there is a relatively easy switch over to next generation sequencing, and vice versa. If next generation sequencing is implemented because of COVID, for example, the similar skills are required for NGS and for Sanger. So I think going forward, both methodologies will go hand in hand, and depending on the situation, the one might work better than the other.
Great. Yes, thank you. Natasha, anything to add, NGS versus Sanger in this setting?
Not really, but I'll reiterate what Kim said. I think in future they both have benefits, and labs will get used to using the one or the other depending on the samples and what they want to get from it.
Yeah, and I think it's definitely true that with all of the additional sequencing that's going on as a result of COVID, labs are definitely better set up to perform both approaches at this point. From an extraction and amplification standpoint, whether it's Sanger or whether it's NGS, there's been a general increase in capability, I think, not just in LMICs, but across the globe in terms of being able to perform sequencing. So I think at that point, I'd like to thank you, both for your participation in this roundtable and also for your excellent presentations. And perspectives on HIV drug resistance surveillance. And I'll hand it back to the moderator for a closing comment. Thank you.
Thanks, Steve.
Thank you.
Thank you again, Dr. Steegen, Dr. Wood, and Dr. Williams for your time today and your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.